some of the work that I'm already doing. So let's just get right into it. So here's just kind of an overview of some of the areas that I'll be touching on. I'll be first talking about traditional epidemiology of coffee and health, then very briefly the concept of systems epidemiology, and then getting into much of my work on the genetics of coffee and caffeine intake, followed by how we can actually apply some of the information that we're gaining from the genetics research of coffee into doing better research on coffee and health. And finally, if there's time, some of the other work I'm doing in other omics for coffee. So if you don't know, this is, coffee is one of the most widely consumed beverages in the world. And here's just kind of a global kind of look at the consumption of coffee. Obviously, the big consumers of coffee are really in the Nordic countries, such as Finland. They're consuming quite a bit. Their average is maybe 4 cups per day. For North American countries, it's largely about 2 cups per day. But as you can see, it kind of varies. And overall, the intake of coffee is quite widespread. The Asian countries are, again, still big tea consumers, but coffee is on the increase for those populations as well. So given the widespread consumption of coffee and also the availability, it's not like cigarettes where it's illegal restrictions. Coffee is widely available. And given that, there's been great interest in understanding the role that coffee might play in health. And here's just kind of an overview of the published meta-analysis. I won't list all the studies that have actually looked at it, but this just kind of highlights the fact that there's been great interest in looking at coffee and health. Some of these meta-analyses have included close to, for example, pancreatic cancer. 54 studies were included, so you can imagine there's been a history of epidemiological studies of coffee and health. So looking at the direction of effects, we see that coffee has had mixed relationships with these diseases. The most consistent findings, however, have shown that coffee reduces type 2 diabetes and reduces Parkinson's disease. But for other outcomes, such as various cancers and even cardiovascular disease, the relationship between coffee and health based on epidemiological studies is quite unclear. Reproductive health, there's really limited types of studies that enable us to look at coffee and health. It's very much confounded, and I'll get into some of the issues for limitations to epidemiological studies in the next slides. But despite the overall clear understanding of the role that coffee has in health, only just recently, in 2015, the dietary guidelines in the U.S., where I'm located, have actually included coffee for the first time in their dietary guidelines. And those guidelines, they say, the old guidelines, as I mentioned before, they did not even address coffee. The new guidelines say that up to 5 cups of coffee per day is safe. Obviously, some people have used these guidelines as promotion to increase their intake. Like, people really look at the guidelines. It says up to 5 cups of coffee is safe. We're not encouraging you to consume more, but if you're already consuming 5, then it's probably safe. Now, I looked into the somewhat equivalent, I believe, for Europe, and they also are pretty much consistent with what the U.S. is doing, in that habitual intakes of up to about 400 milligrams of caffeine. Now, again, this is looking at caffeine, and coffee is actually one of the major sources of caffeine. And a cup of coffee per day contains roughly, and I say roughly, and I'll talk about it later, about 100 milligrams of caffeine. And so this equates to about 4 cups. And so the guidelines here suggesting that 4 cups of coffee per day is safe is similar to what we're seeing in the U.S. For pregnant women, again, the literature on that is really inconsistent, quite limited. And so they say that really women should be consuming or should limit their intake of caffeine to that, which is equivalent to about 2 cups of coffee per day. And here's just a more recent snapshot of the pregnancy. The WHO looked at the literature looking at caffeine or other dietary compounds with pregnancy. And in 2016, they reviewed the literature, and much of the evidence where they talk about restricting caffeine intake is based on, looking here, is based on the epidemiological studies, which we already know are somewhat inconsistent. They're also considered some clinical trials, and I kind of clicked on here to see what clinical trials they were looking at. They included one, and it really had no relationship to pregnancy outcomes. Actually, this clinical trial was probably assessing preterm apnea, which is where they use theophylline as a bronchodilator to help these the preterm infants, and it's actually been shown to be quite effective. So how this actually relates to pregnancy outcomes, there's no connection. So really, any guidelines that they provide is based on epidemiological studies. And it's kind of ironic that just only in 2016 has coffee been actually dropped from a potentially carcinogenic to unclassifiable. So up until this year, coffee was potentially carcinogenic. Yet the 2015 guidelines went on to say that it's pretty safe. So I'm not sure how these were kind of how this change was motivated by that work, but looking at how the review went about, you can see that the 2016 changes was largely weighted on the prospective cohort studies of coffee and cancer primarily. So based on some of the guidelines that we're seeing, you see that a lot of the work that's coming out in terms of public health implications are really based on epidemiological studies. And so it's important to highlight some of the key limitations of this study design. Classical limitations are the measurement error and confounding. Measurement error, for example, might be the size of a cup that are typically captured in, say, a food frequency questionnaire. Although food frequency questionnaires actually do capture coffee quite well, there is still obviously some measurement error in actually what constitutes a cup. People talk about, I'll have a mug, but how big is your mug compared to someone else's? Likewise, if there's interest in caffeine, the caffeine content of some of these products out there have actually changed. And here's just one example that was recently out for sale, and it's coffee which produces for each cup of coffee there's about 702 milligrams of caffeine. So that's roughly 7 cups of coffee in 1 cup of coffee. So if epidemiological studies are looking at 1 cup of coffee, if we have tons of people consuming this kind of coffee, then that really makes our research a little difficult. And in terms of confounding, here's just kind of a snapshot of some of the characteristics of heavy coffee drinkers versus non-coffee drinkers. And these are based on U.S. cohorts that I work with quite often, and also the UK Biobank. And you can see that some of the correlations between the coffee consumption and characteristics kind of vary across populations and even by gender. But generally, heavy coffee drinkers are more likely to be smoking. They're more inactive. They're consuming more alcohol, saturated fat, red meat, less fruits and vegetables. And obviously, these are risk factors for other diseases. And so you see that there's obviously potential for confounding. And not all of these studies have this kind of information. Other challenges, again, very similar to measurement error, is that coffee is complex. That even a 1 cup of coffee can vary in its composition based on the type of beans that you use and even how it's prepared. What I've observed here in Bristol is that when I went to Sainsbury, you guys have a great aisle of instant coffee. Like, oh my gosh, we don't drink that much instant coffee in the U.S. U.S. is all basically brewed grounds, and so our coffee aisle is quite different from yours. You guys have got a great selection of instant, so anyone who's interested in instant would definitely enjoy coffee here, I'm guessing. But there's maybe a few products for coffee instant coffee in the U.S. So that even makes a difference on the composition of coffee. Individuals also vary in their physiological exposure and response to coffee, and I'll go into that in detail here. By exposure, I mean by absorption, distribution, metabolism, excretion, classic pharmacology. One example being the caffeine content of coffee. The half-life of caffeine can vary between individuals about 2 to 12 hours. That's a big gap. The physiological response to caffeine is largely due to the ability of caffeine to block the actions of adenosine at the adenosine receptors, predominantly the A2A receptor and A1. And by blocking the adenosine receptors, it gives rise to a number of physiological effects, often those which we equate to caffeine, such as the psychostimulant effects. But besides the psychostimulant effects, there's also other effects, such as impact on insulin and glucagon release, and also vasodilation, some things that we don't necessarily feel directly, but this is what's going on indirectly. And finally, epidemiological studies do not provide insight to causality or the mechanisms by which coffee contributes to health. But in the past few years, there's been a great boom in what we call the great omics, which are high throughput or collective technologies that explore the different relationships of various types of molecules that make up the cells of an organism. Long story short, they're high throughput, basically looking at everything at any particular level, and provides a global view, and also underscores potential for hypothesis generating. And much of my experience has been with the genomics area, and I'm working towards branching out to metabolomics, proteomics, and possibly transcriptomics. So with these new technologies, we have an opportunity to go from our traditional black box epidemiology to what I've coined as systems epidemiology. Others might call it some other thing like systems biology, but if we're looking at epidemiological designs, I would call it systems epidemiology. And it basically breaks up that black box into its different levels. Obviously, there are so many other omics going on, but these are just examples. And it actually now takes into account the between person variability. And by doing that, we provide insight into mechanisms. And also, by looking at the different individuals' responses, there's great potential for informing nutritional guidelines, providing novel preventive strategies which target certain subgroups of the population. And there's this great goal for personalized nutrition. Obviously, this is probably much more, not so much in the near future, but providing insight into mechanisms is really where we're at now. And I think that's we're making great progress in that. And so in the next few slides, I'll be kind of summarizing where I'm at in terms of the genomics of coffee intake. So much of the work that I'll be presenting to you is really in collaboration with the Coffee and Caffeine Genetics Consortium. And here is just obviously the globe, just overlaying our coffee consumption group. And the black dots are just the acronyms for some of the studies that are contributing to. There are obviously so many studies. There's roughly 30 studies contributing to this group. And as you see, most of them are spread out in Europe, as well as the US, and some in Australia, and one in Pakistan, I believe. So our first efforts in applying this omics or genomics to coffee consumption, we conducted a genome-wide association study of caffeine, which was published a couple of years prior to coffee consumption. So the results we present today are based primarily on the GWAS of coffee consumption, because it's based on a larger sample set. And those of you who are not familiar with genome-wide association studies, basically we look at all the genetic variants in the genome and look at associations between those genetic variants and your outcome of interest. In this case, we were looking at cups of coffee consumed, and we were restricting our analysis to coffee consumers to potentially get more of a dose-response effect. And this is just your classic Manhattan plot, showing you each of these dots correspond to a p-value. Each of these dots correspond to a p-value for an individual SNPs. And there are approximately 2.1 million SNPs here plotted. And the points in green are loci that reach genome-wide significance. So our most significant loci were mapped to genes that were related to pharmacokinetics, so the metabolism or absorption. And I'll just go into the detail of some of these. CYP1A2 was a very plausible candidate. And that what's interesting is that we actually know quite a bit about caffeine metabolism. And here's just kind of a breakdown of caffeine and all its metabolites. And CYP1A2, highlighted in the red here, is responsible up to 95% of caffeine metabolism. So what we're finding here is that this gene related to caffeine metabolism is also linked to coffee consumption behavior. AHR was another highly significant locus, and obviously another biological candidate in that it regulates the expression of numerous CYPs, but specifically CYP1A2 and CYP1A1. So a biological candidate. P450 oxoreductase actually has an important role in the function of CYP450s, including CYP1A2, another biological candidate. ABCG2 is a classic drug transporter, and it functions at various tissues in the body, the brain, as well as the liver, kidney, and intestine. And so it's probably playing a role in transporting caffeine, although there's only been a limited amount of literature looking at that, but it's quite possible. In addition to these candidates involved in caffeine metabolism or its absorption, there's also ones related to its response. And these are actually the ones that I expected, given that we were looking at a behavioral trait. We found brain-derived neurotropic factor, and also the gene, and SNPs close to the gene mapping to the serotonin receptor, serotonin transporter, I believe. And so these are the ones that I actually would have expected to be up higher. I didn't expect the ones involved in metabolism to be the ones related to behavior. Ones which really remain questionable are genes or SNPs that related mapping to GCKR and MLX-IPL. The SNPs that we found were actually ones that were previously linked to glucose homeostasis and also lipid levels. So how it relates to coffee consumption behavior remains quite a bit of a mystery. So since that work, I've actually been working with a subset of these populations that actually have metabolite levels of caffeine. And that gave us the opportunity to conduct GWAS. Ideally, I would have wanted to do this first, but at the time, this type of data wasn't available. What's interesting is that the results were kind of overlapping with what we found with the coffee consumption behavior. But just to begin, the metabolites of interest were those that I have underlined here. In addition to the individual metabolites, I also examined a ratio of paraxanthine to caffeine, which is actually a clinical measure of caffeine metabolism and also a marker of CYP1A2 activity. Caffeine is actually a probe drug for this particular enzyme, so it kind of made sense to give this a try in a genome-wide context. I just highlight again that these are cross-sectional, so we weren't actually giving people caffeine, so we really don't know if they were consuming caffeine at the time. It's just cross-sectional studies of caffeine metabolites in the plasma. And the findings were very similar to what we saw with coffee consumption behavior. Our top hits for looking at the caffeine metabolite, CYP1A2 and AHR, which, as I mentioned before, I expected. Interestingly, we found, well, this wasn't so interesting, but it was nominally significant. So it's possible that, given the sample size of 9,000 relative to our coffee intake GWAS, which is based on over 90,000, we may just been underpowered, but it was nominally significant, so just below the negative minus 6. ABCG2 was also nominally significant. What was interesting is the GCKR was still up there too. So to my knowledge, or to anyone's knowledge, the role that GCKR might play in caffeine metabolite levels is questionable. And actually, with our paraxanthine to caffeine ratio, which, as I mentioned, was a potential marker of caffeine metabolism, CYP1A2 activity, we found even stronger signals. Again, CYP1A2 and AHR. Interestingly, CYP2A6 came up. Those of you, obviously, in this group might know that CYP2A6 is a key gene involved in nicotine metabolism. And so that was kind of an issue. We were actually, in our models, we were adjusting for smoking. But looking a little closer at the, these ones were nominally significant. But I was really still interested in this particular one. And so I kind of did a little bit more investigating. And although we were looking at the paraxanthine to caffeine ratio, CYP2A6 has not been shown to play a role in this particular pathway. But what's interesting is CYP2A6 actually works downstream, and that it's involved in metabolism paraxanthine. So what we're seeing with the paraxanthine to caffeine ratio might just be an indirect result of this metabolism. And interestingly, we found that paraxanthine levels were also increased with this variant, suggesting that what we're seeing here is potentially impaired paraxanthine metabolism rather than caffeine. So again, we're seeing some impact of this on caffeine metabolism. And so for those of you who are kind of getting confused, or one thing I didn't talk about was the direction of these effects. And so here I have a summary showing you the genes that we found related to plasma metabolites and also behavior. So this is a kind of a key column that's quite different from these ones here. So what I show here are the directions of effects. So the red indicates increase and the green is decrease. And so all are with reference to the same allele. So looking at, say, AHR, for example, the allele that gives rise to higher levels of caffeine also give rise to lower consumption of coffee. At the same time, that same variant is linked to lower caffeine metabolism, so suggesting impaired caffeine metabolism. So taken together, this would suggest that individuals with impaired caffeine metabolism would have higher caffeine levels if they consume coffee. And having those high caffeine levels will likely impact their behavior, so that they naturally reduce their consumption. So it's kind of interesting that we show this here, that we're at a population level. These genetics are helping us titrate our levels of caffeine so that we get the optimal, which I assume is to be the stimulant effects of caffeine, because that's something that we are pretty much well aware of in terms of the effects of caffeine. CYP1A2 also showed a similar pattern. P450 oxoreductase, similar pattern as well. ABCG2 is a little different, and actually the combination of this data would suggest a hypothesis by which ABCG2 is related to caffeine. Here we find that metabolite levels are actually higher with the variant, and individuals are actually consuming more. This might suggest that ABCG2 might be acting at the blood-brain barrier, so that we're consuming coffee, caffeine levels are going up, but the caffeine isn't getting into our brain. And it's because of some sort of impaired transport of caffeine. So we just keep drinking. And so that could be potentially detrimental. Right? If we show that caffeine is toxic, for example, we got some people with this particular variant that are just going to keep consuming and just not feel the effects. GCKR was interesting. It also followed a similar pattern to AHR and CYP1A2, including the impact that it has on ratios. So we can't discount the potential role that GCKR has in caffeine metabolism. CYP2A6, you're going to see it obviously here. When we looked at this particular SNP in the UK Biobank, at that time we had a much greater sample size, and we actually showed that this variant was actually linked to coffee intake behavior. In the genome-wide association study, which I showed you earlier, coffee intake, it was nominally significant, and so I didn't show you that CYP2A6 might actually impact behavior as well. Ones that show no evidence of a relationship here were those with BDNF and serotonin transporter and MMX-IPL, because it's linked to a reduction in coffee intake with no impact on metabolites. This would just suggest that these might be acting through non-caffeine pathways, so possibly behavior. So this is kind of a good example of how we can use information for both studies to provide potentially new hypotheses worthy of further investigation by using the combination of them. So how can we use these genetic findings? Obviously, when I first started this work looking for genes, a lot of the issues were, okay, found these genes, now what? It's not like they're predicting disease. But now we have an opportunity to study the effects that coffee has on health much better. And what I've been focusing primarily on up to date was really looking at gene-coffee interactions in health. And my visit here has really been to get some more insight into how we can effectively use this data for good Mendelian randomization studies. And just an example for gene-coffee interactions in health, I'll just take you back to my earlier work as a PhD student. At this time, we were working with candidate SNPs, so I was actually genotyping, doing the DNA extraction and electrophoresis really before we had these genome-wide chips. So I was actually in the lab just doing thousands of genotypes. And my key hypothesis of my thesis was that if coffee is associated with particular outcomes, it might be modified by our ability to metabolize caffeine, if in fact caffeine is a component that is linking coffee to some of these outcomes. More specifically, I was genotyping for CYP1A2. So even back then, CYP1A2 was an obvious candidate. Obviously, the variant that I was looking at wasn't exactly the one that we found in GWAS, but it was still the same candidate gene. And so I was interested in looking at variation in this gene and the relationship between coffee and coronary heart disease. And this was in collaboration with a group in Harvard. And this is just kind of a snapshot of the basic traditional epidemiology that I conducted. And for this, I had about 2,000 cases of myocardial infarction or heart attack and 2,000 controls. And in this population, we found that consuming 4 or more cups of coffee per day was associated with about a 1.5-fold increased risk of myocardial infarction relative to those consuming less than 1 cup of coffee per day. However, after genotyping and then using that data and stratifying the population by genotype, we found that the increased risk was only among those with the impaired activity enzyme, so impaired caffeine metabolism. So we found that a significantly increased risk already with 2 or more cups of coffee per day. We found no significant increased risk among those with rapid caffeine metabolism type of genotype. And what's interesting is that we actually found a potential protective association between moderate coffee consumption and myocardial infarction. And there have been some epidemiological studies suggesting that it has potential protective effects. This might suggest that individuals who are rapid caffeine metabolizers might be able to reap the benefits of other potentially protective compounds found in coffee. Because caffeine has been cleared from the system, maybe those health effects become unmasked once the caffeine is gone. And so that's just kind of provides some insight into how we can use these genes and understand the role that coffee might play in health. This might suggest, because we found an increased risk, might suggest that caffeine does have an adverse effect on heart disease. There's also interests that are probably more applicable to this group. There's some interest in determining whether caffeine is a potential drug of dependence. Now, according to the latest edition of the DSM manual, it's not currently suggested that caffeine is addictive, but we have what we call caffeine withdrawal, and that it's a syndrome resulting from abrupt cessation and reduction in caffeine following prolonged daily use. So it's not dependent, but there's this caffeine withdrawal syndrome. And so given that we have the genetics, this genome-wide association study for coffee behavior, and also other GWAS of alcohol intake, smoking, and also more recently cannabis initiation, a group not involved in this work, urine actually in this study was involved in this work. It's kind of a witty, very simple but witty experiment. They looked at the genetic correlations among all these different potential substance uses. And just looking at these correlations by using linkage disequilibrium score regression, they found, here's just the kind of the matrix looking at the correlations. It's not so clear, but here we have coffee consumption per day, so that's based on the GWAS of coffee intake, which I showed you. And they looked at the genetic correlation with other outcomes. And what's interesting is that we found a strong correlation with cigarettes per day, and significant, but a little bit less strong for alcohol consumption and others smoking behaviors. And so that's interesting in a way to apply some of the knowledge that we're gaining from genetics and other confounders. Also the Mendelian randomization, which leverages the random allocation of genotypes at birth and using the genetics as a non-confounded marker of our risk factors of interest that's independent of the outcome specifically, but only through the risk factor. And this actually, just recently, there have been quite a number of efforts to apply Mendelian randomization to coffee consumption. And here's just a very kind of summary table of some of the recent MRs. And I won't get into details. The methods that they've used were quite different. The IV or the instrument variable that they use for coffee were generally the ones from the coffee intake GWAS, but they also varied in the size, the number of SNPs that they included. If you recall, if I showed you that the table looking at the kind of the intersection between the coffee intake and metabolite GWAS, with some of these SNPs have very different functions. And for studies that are using, say, 8 SNPs, using kind of a mixed bag of different genes related to coffee intake, we really don't know how to really interpret any results that come out of this. But at that point aside, most of the results have been rather flat in that they haven't found any support for causality linking coffee to health. Perhaps it's because they haven't used the optimal instrument. Maybe the methods are slightly different. The only one that actually has panned out with greater efforts and better design and bigger sample size, which is always a big issue for Mendelian randomization, has been this one looking at smoking heaviness. And they found that strong support suggesting that higher cigarette consumption causally increases coffee intake. What's interesting is that there's actually a lot of biology that would support this as well. What's interesting is that the compounds found in smoking actually induce CYP1A2. So smokers actually metabolize caffeine more quickly. That's probably why they're consuming more. And so this is almost like a proof of principle, an independent proof of principle analysis, and it actually supports the hypothesis. And obviously, most of the work that I've been doing has been applying these to doing better studies on coffee and health. But obviously, everyone's interested in see how we can use this in the clinic. Can we use this for potential risk stratification or even personalized coffee recommendations? It's possible, but I think we still need to do some additional work. Because for example, my gene coffee interaction only looked at, say, CHD. Maybe caffeine specifically has protective effect, and there's some experimental data suggesting that it is protective for, say, Parkinson's disease. And they've actually done, say, adenosine receptor type work, and there's actually drugs that are actually targeting the adenosine 2A receptors based on some of the work we found with caffeine. So even if some person is metabolizing caffeine quickly and thinks that, oh, I'll be fine to consume lots of coffee, or individuals who think they have impaired caffeine metabolism think that, okay, I should avoid coffee. Well, it might also depend on the family history. If they're in a family that everyone has Parkinson's disease, I wouldn't suggest that they cut back on the coffee. I would definitely just, consume enough that you're actually getting exposed to caffeine. This is just kind of a demonstration of personal recommendations. This person, obviously, they've been told to consume 1 cup per day, and they're obviously consuming 1 cup per day, but again, their cup of coffee the size of their cup of coffee is different from someone else's. And there's time. Obviously, to date, most of the research that I'm showing you looking at coffee has really been pinpointing, or everything is always looking at caffeine. So what other compounds in coffee might be protective? So coffee is complex, as I mentioned. So what about these other compounds? How can we effectively assess the relationship between these coffee compounds and health outcomes in epidemiological studies? And so this is where other omics come in. And just recently, I've been kind of looking at metabolomics, and more specifically, kind of moving away from epidemiology, kind of population-based epidemiology. I have the opportunity of working with data from a coffee trial. This was already published. It's a Finnish trial, actually, where they asked 47 participants to abstain from coffee for about a month, and then they withdrew blood. Then they were asked to consume 4 cups of coffee per day for a month, and then they were asked to consume 8 cups per day for a month. Now, this work has already been published, and they found that coffee had some benefits on adiponectin, no impact on glucose levels. Just really somewhat disappointing because they didn't find a whole lot, but they are also looking at clinical measures of metabolic related to metabolism. And so having having this blood drawn and stored, I contacted the PI and said, hey, can I use your samples, see if I can profile them? And so he was happy to collaborate. And with that, I had these samples and I had them profiled. And I worked with Metabolon, which is a commercial company who's expert in metabolic profiling. And with their platform, I was able to profile just over 700 metabolites. And I'm not obviously listing all 700 metabolites, but these are the different pathways and kind of subgroups of metabolites that we assessed. So they capture various different pathways from xenobiotics, also including our drugs, so it also included our caffeine metabolites, but also other ones related to amino acids and lipids. So it's very diverse, and that's the idea of this platform, is kind of a discovery assessment. And it was actually very successful in that we found 115 metabolites linked to coffee consumption, accounting for the trial itself. And not surprising here, obviously I'm not listing all 150 metabolites, but these are the pathways. I'll just focus in on one or two pathways that were enriched with metabolites. Most of our metabolites actually increased with coffee consumption, and many of these were related to xenobiotics, and one specifically would be xanthine metabolism. So all our caffeine metabolites went up. That was expected. Oh, well, that's not fun. Was there anything new? Benzoate metabolism metabolites went up, and these metabolites are actually a product of our gut metabolism. And we do know that the chlorogenic acid component found in coffee is not metabolized by us specifically, but rather by our microorganisms in the gut. So that was interesting to find. We also found ones that were linked to steroid metabolism, also fatty acids. What I found especially interesting was the endocannabinoid system. These metabolites in this pathway actually went down. And endocannabinoids are often related with cannabis use. And it's never really been linked to coffee consumption. What we're showing here is that at high consumption of coffee, these levels are going down. They especially went down with 8 cups of coffee per day. And looking at some of the literature suggests that endocannabinoids typically go down in response to stress. And so it's their way of trying to bring your body back into equilibrium. And so it might be that although in our finished group, 4 cups per day was what they were used to, 8 cups per day might have just been tipping their scales in terms of stress. And so that might have imposed some stress, and that's where the endocannabinoids went down. Whether this goes on to translate to habitual intake and that our endocannabinoids are going down remains to be known. But we have these population data and with metabolomics that also have measured endocannabinoids. It'd be interesting to see if those go down as well. This system is especially interesting because it's been linked to a number of different pathways and even health outcomes. For example, it's the endocannabinoids have functions in appetite as well as liver health and other pathways. So it's never really been, endocannabinoid system has never been implicated in the relationship between coffee and health. But here we have a potential pathway that maybe it is meeting the effects that coffee has on certain health outcomes. And so that's just kind of a new ideas and just kind of highlighting the hypothesis generating component of metabolomics and kind of pushes the science forward. And for this data, I also have the complex lipids platform that's done by Metabolon as well, and also proteins. And this data I'm just sitting on, I haven't had a chance to look at yet, but it'll be interesting to follow up to see if we find anything new. The proteomics I was especially interested in because based on this is work that has just recently been accepted for publication in Journal of Internal Medicine, where I collaborated with a group in Sweden where they did a proteomics of their population participants. And they found in a cross-sectional design that we found that leptin, as well as this novel protein, chitinase-like protein, which I believe is also YKD40, these changed, they actually went down, they were negatively correlated with coffee consumption population level. And so, again, because this is just cross-sectional analysis and an epidemiological study, we don't know if it's causal, it could just be something else. But because I now have proteomic data that includes these proteins, I can actually look at that in our coffee trial to see if I can show that. And then also bringing another layer of genetics, we know that there are genetics for leptin, and there's one study that actually did like a genome-wide association of all these proteins. And so we'll actually have potentially new instruments for these proteins to look at potentially MRs or gene coffee interactions. So again, it's kind of a cycle, but instead of focusing just on caffeine, we'll have an opportunity to look at other pathways implicated by caffeine. And in conclusion, these are just the people that really been helpful in some of my research, obviously collaborating, providing the data, having these analysts doing all the analysis, much often on their kind of like a hobby. It's just things they have to do on time. Obviously, their biggest interest has not been in caffeine, but they're very interested in collaborating. At the same time, with these genetic consortium, you take some and you give some, and that's just the way it usually goes around. And these were my postdoc supervisors: Frank Hu, does a lot of primarily nutrition. Peter Kraft, he's a genetic statistician. I've learned much of my work in the GWAS with him. I continue to collaborate with Peter Kraft on numerous projects. And Ahmed El-Sahami, who really, he was my PhD supervisor. Prior to working with him, I had no idea what a gene really was. And so that really, he provided me the foundation for much of my work today. And it's interesting that my PhD was on coffee, CYP1A2, and health. And 10 years later, I'm still working in the area. So I think I've just been building on that work. And so obviously, all my work has been really on coffee and caffeine. And as much as I try to shake it, I just keep coming back to it. So I've decided that that's just going to be my future and research. And with that, I'm open to any questions you guys might have.